Okay, so we're going to get started with all you lovely people who are here. Good morning, Zyla. How you doing? All righty. Up, here comes another. All right, so we're going to go ahead and look at genetics. And we're going to do kind of a refresher on DNA and everything that we did last semester in terms of uh, genetics, DNA structure, all that sort of thing. So I want you to go to Canvas and I will show you which we are on. We're going to start the unit. Here we go. We're going to start the unit on genetics. My screen is not cooperating. All right, so find this unit. Oh, let me make sure that it's published. I don't think it's published. There we go. All right, there we go. It's published now. So you might have, you've already gone in, you might have to refresh your page. Let me view this as a student. And there we go. Okay, modules. The student view. Okay, it was published on my side, but let me know if it's not published on yours. Because when I was just now looking at the student view, I didn't see this. This is what you should see. Good morning, Landon. Happy New Year. It's not. So let's see what's wrong with this. Uh, okay. All right, I had an extra button to push. Try it now. There you go. All right, so this is what you should, everybody should be able to see it now. If you, okay, thank you, Natalie. All right. So as you can see in the picture there, they are holding DNA molecules. So a lot of what we talked about last semester with DNA structure and bonding and matching pairs and all that sort of thing. We're going to kind of revisit it a little bit and um, tie that into um, why things look like what they look like. So we have with this we have a lot of activities to do because you have to learn how to match up um, different characteristics. You have to predict characteristics. And then there's a lot of vocabulary. So what I would like for you to do for the next few minutes is just give this vocabulary list a good look over, and then we'll dive into a Nearpod and look at some of these. But just take a look at some of the vocabulary. And as you see, it's quite an extensive list of terms. But take a few minutes just to look that over. Okay. All right, everybody's in. All right. And this semester, we want to make sure everybody stays on camera. Um, I got a couple of housekeeping rules. You got to stay on camera. You got to stay focused, stay on task, and stay current. We don't want a whole lot of making up and things of that nature so that you can get full credit for everything you do. Like I said, at the end of this semester, by May, probably maybe April, you have an end of course test to take in biology. So at some point, we'll stop and do a review packet that covers all of the things that we've done for the year. Um, to tie everything together so that you can be most successful. So don't throw any notes. If you're taking separate notes, if you have separate worksheets, if you have a folder that you kept last semester's notes in, don't delete it. Put it off to the side because everything you did last semester, you're going to need for the end of course test at the end of the year. And this is the state end of course test, not the class final exam. This class has an end of course test. Okay. All right, so I'm going to stop sharing for a minute and you all look at the vocabulary. I'm going to change screens. Okay, So I put the link for the Nearpod in the chat. I am going to do it in teacher-led mode. But do log into the Nearpod so that you can respond to the questions. Okay, according to this, I only have 4 participants. Everybody needs to log in, please. Who we got? Who we got? Who we missing? Got Malik. Maxwell. Wake up, Maxwell. Okay. All right. Okay, I'm going to start. Maxwell, wake up.
Genetics helps us understand the biological programming behind all life forms. But what exactly is the science of genetics? And what does its future hold?
All right, so automatically it's asking you to participate. So like I said, we did a little bit of DNA study last semester, so you know a little something. So I'm going to change this screen over and give you a few minutes to answer the question. How much do you already know about genetics? Oh, again, we need participation from everybody. Max, wake up. I said, all of you know a little something about genetics. So let's talk about what you already know. All right, so I'll give another minute before I switch over. Got no answers from some people. Let's look at the ones who have answers. Okay, Piper says genetics studies traits of organisms, and these traits are inherited directly from family members, like our parents, aunts, uncles, etc. Kishan says, I know genetics is DNA that our parents, and they make up who you are, and that's good. Malik, the field to predict future outcomes. Okay, future outcomes in living things. Okay, because we're going to talk about non-living things don't have genes. All right, genetics is a study of how traits and characteristics are inherited from, okay, they predict how you look. They study of human characteristics from, okay, good. All right, thank you. Maxwell, where'd you go? Log in, please. All right, we're going to go on.
Genetics is the study of heredity, the expression of traits and how they are passed from generation to generation. For thousands of years, humans have observed this inheritance of traits and implemented their knowledge to breed and domesticate plants and animals. However, the science behind inheritance was only starting to be understood in the mid-19th century. Around 1865, Austrian monk and botanist Gregor Mendel published the results of his hybridization studies of pea plants. In his findings, he noted the role of factors that influence the expression of traits. These factors later became known as genes.
Okay, participation. That for now. All right, who discovered this? Now you're going to find that in every branch of science, there's always somebody who is the father of. So knowing who the father of genetics is an important thing. Because we're going to talk about him a lot and some of his rules. He has a lot of rules. All right, how we doing? Yeah, I don't have. There we go. Everybody has answered and everybody has said Gregor Mendel. Excellent, excellent, excellent. All right. So yeah, so remember his name because it's going to come up a lot because he's kind of the head man in charge of genetics.
Each human has between 20,000 and 25,000 genes. This collection, called a genome, determines a person's traits by influencing factors on a cellular level. Genetic information is stored in every cell's nucleus. Structures called chromosomes carry this information in the form of deoxyribonucleic acid, or DNA.
All right, so we got another question. And what I find is with these questions is they, just before they ask the question, it's kind of a test to see if you're listening. If she just answered it just before the question. Okay, in the nucleus. All right, so we've talked about the nucleus before. We have talked about cell structure. So we are still going to kind of touch on cell structure a little bit, the nucleus in particular, because all of the stuff that we're going to talk about is going to happen in the nucleus. But because it's in the cell, we do need to kind of briefly go back over some cell structure a little bit.
DNA is a double helix of nucleotides, chemical compounds composed of sugar and phosphate molecules, along with the bases thymine, adenine, guanine, and cytosine. These segments of DNA are what we call genes, and it is within those genes that chemical compounds provide the coding for all information about a person's inherited traits. Human cells contain so much DNA to carry this large amount of information that, if unraveled, the DNA in each cell would be over 6 feet long.
Okay, question. What is DNA, or deoxyribonucleic acid? Okay. Okay, we still have somebody who hasn't answered yet. All right, so the letter A is correct. If you haven't answered yet, go ahead and select A. If there's somebody hasn't answered. All right, a double helix of nucleotides. So again, we got to keep all these vocabulary words in mind. So double helix means that it's double stranded. The nucleotides are our units, our base units that consist of a phosphate, a sugar, and at least one of the four bases. That makes up a nucleotide. Okay.
At the turn of the 21st century, an international effort to decode human DNA was launched. Called the Human Genome Project, it ended up identifying about 99% of the entire human genetic sequence.
A revolution in medical science whose implications far surpass even the discovery of antibiotics.
All right, true or false? An international effort to decode the DNA was launched by a Human Genome Project. It ended up identifying 99% of the entire human genetic population. I mean, sequence. True or false? Let's go. Everybody. Maxwell, I don't know where you went, but come back. Are we still missing somebody? But yes, that's true. And what they mean is that they have looked at every possible genetic characteristic that exists in human beings. So this is the Human Genome Project. Now, all living things have genes. So while they have studied them, they particularly focused on what are all the different kinds of traits that humans have. And it's very vast.
Discoveries in genetics research have unearthed tremendous opportunities in medicine, such as genetic testing and the manipulation of genes. But with these opportunities come risks and ethical questions, and finding the answers to those questions may be the next stage of our understanding of genetics.
All right. So before we start the unit, take a minute to think about what are you curious about as far as genetics are concerned. You said you know a little bit, everybody knew a little bit about genetics that when we started. What don't you know? We had a quick look at the vocabulary list. What don't you know that you think would be interesting to know? And if everybody answers all at the same time, then we get to move on. All righty. So we got just a couple of answers coming up. All right, after reading through the vocabulary, I remember most of the material. Okay. All right. That's an interesting word, so we'll make sure that we look at that. Everybody gotta have an answer. So you gotta be curious about something. So no answer is not an answer. All right. Just learn more about genetics, different species, and we are definitely going to do that. And we are going to look at exactly how do a little bit of discussion about that genome project. In fact, let's see, what time is it? Okay. All right, Kashawn, are you with us? Are you with us? What you curious about? All right, we are definitely going to spend time manipulating genes because there's a device called a Punnett square, and it goes from being 4 to 16. And so we're going to spend a lot of time learning how to manipulate genes and looking at outcomes, predicting what things will look like. I even have an alien project that you you're going to work on, where you're going to predict what your personal little alien is going to look like. Okay. All right. Thank you. Alrighty. So we will come out of that. All right. So let's look back at Canvas. Let me stop my Nearpods because it's still singing. Stop. Okay. All right. So let's look at the first page. All right, so first person you see is Mendel. And so we looked at the fact that Gregor Mendel is the father of genetics. And why is my page not loading? There we go.
Created using Powtoon. Created using Powtoon.
Okay, so let's look at this. I'll take a few minutes to read that over. I don't want to read it to you, but this is a little background information. So most of you talked about, you're familiar with the term traits, and those are the characteristics that we get from mom and dad. You get, and when we're going to talk a lot about meiosis, because meiosis, remember, it separated the characteristics, and then when the egg and sperm came together, they gave half of the characteristics from mom, half of the characteristics from dad. So we are going to kind of revisit meiosis a little bit to just refresh our memories as to what that's all about, because that's how we get a combination of traits. Okay. So take a few seconds to read that over. Okay, so the first vocabulary word on this page is selective breeding, also known as artificial selection. That means that you've made these selections on purpose. They didn't happen by accident. You specifically you picked an organism with a certain set of traits, and you combine them with another organism with another set of traits so that you can create the output that you're looking for. So it was not natural, okay? Natural selection, and this is selective breeding. So know the difference between that. All right, so we're going to look at
Genetics Primer, Mendel and Modern Genetics Terms. Gregor Mendel is known as the father of modern genetics. Mendel was an Austrian monk, and he lived from 1822 to 1884. His genius was that he noticed differences in peas being grown in the monastery's garden and applied the scientific method to determine the cause of these differences.
Okay, so one of the things that you need to, I'm gonna pause it.
Mendel chose the garden pea
Can't pause
Pisum sativum because
it. Let him talk.
peas were easy to grow. They had a short generation time. They had a number of contrasting traits that were easy to distinguish, such as short versus tall, or white versus purple flowers. And they also normally self-pollinated but could be cross-pollinated. Mendel was able to control mating this way by removing the male or the female flower parts and cross-pollinating with a paintbrush. Normally peas do cross-fertilize or fertilize themselves, but he didn't want this to happen. Mendel didn't really know about inheritance. No one had great ideas about it at that point in time. We knew what cells were, but we did not know about organelles, protein synthesis, or even genes or chromosomes yet. All Mendel had to go by was physical traits and his knowledge of flowers and statistics.
Okay, it advances when it wants to advance. Okay, what you do need to remember about Mendel is what he studied. He studied pea plants. He was a monk, and they grew plants in their at the monastery, and he was a scientist as a monk. So he studied peas. Now notice some of the terms that he looked at. He looked at their characteristics: short versus tall, and white versus purple flowers. So these, he was curious as to how do I get these different traits to show up? Okay. Now, we might need to go ahead and talk about reproduction in plants a little bit. So we'll come back to that. But basically, the male structure on the plant has the pollen, and then the female structure has as a section where the pollen falls through, and the egg is inside of that, and that's where fertilization takes place. So self-pollinating is when, if you just kind of brush the plant a little bit, the pollen automatically falls into the female structure, and the plant is the flower is pollinated and will produce, and the seed, the pea plant, the peas are the fruit that will develop from that pollination. He didn't want that to happen, so he removed them so that he could control what plant is pollinating which plant, so that he could get the results that he wanted.
Mendel chose easy to recognize true breeding varieties for his experiments, some of which are pictured here. True breeding, or purebred, means that when the plants were bred, offspring maintained that characteristic over time. Therefore, the offspring showed traits identical to their parents and identical to each other.
Okay, so if you are going to see, all the things at the top are the characteristics, the kind of seed, and we'll talk about what these words mean, because eventually we are going to talk about different types of plants. So the form of the seed, was it gray or was it round? Was the skin on the seed coat, was it white or was it wrinkled? Those were two things he was looking at as far as what it looked like on the outside. Cotyledons are the type of seed that when you open it up, if you ever looked at a pea, and a lot of people don't like peas, but if you ever actually squished one and opened it up, it kind of breaks into two pieces. So that's what they mean by a cotyledon. So what does the seed look on the inside? If you shop at all, you know that there are green peas and there are yellow peas, and some, like, again, most young people don't like peas, but he was curious as to what color was it on the inside. Was it yellow or was it green? The flower that it came from, was the flower white or was it purple? The form of the pod that was formed from the flower. Again, the pod is the fruit that develops from the flower. So what form did it take? Was it a full or was it constricted? What color was the pod? Was it yellow or was it green? And the stem of the plant, when they talk about, when you're looking at a plant, the arrangement of the leaves on the stem are this axil. So that's when they say the axil of the pods. He's looking at where they are placed and the size of the stem. How long is it, or is it a short plant or long plant? Okay, so the arrangement of the leaves on the stem and the size of the stem. These are all the characteristics that he studied just by looking at peas, and based all of his genetic research on just these characteristics.
Mendel examined genetics in peas over generations and kept detailed records. For instance, you can see in this diagram that the parent, or the pea generation, are pure-breeding purple and white flowered plants. They were crossbred with each other. This was done carefully by use of a paintbrush to transfer the pollen from the male parts of one flower to the female parts of the other flower. The offspring that result from the pea generation cross are called the first filial, or F1 generation. Then, if members of the F1 generation are bred with each other, their offspring were called the F2 generation, or the second filial generation.
Okay, so here again, some vocabulary that you're going to have to do, because when we are going to create charts, and we are going to look at what are the characteristics of the parents, and the question is always going to be, what does the F1 generation look like? And if we picked two offspring from the F1 generation, what will our F2 generation look like? So these are terms that you're going to have to become familiar with, and we're going to be working with those as we go along. Oops.
Let's take a closer look at this same experiment. In the P1 generation, Mendel uses his paintbrush to control pollination. The true-breeding white-flowered plants are bred with the true-breeding purple-flowered plants. The F1 generation that results from Mendel's cross all have purple flowers. Then Mendel allowed the F1 generation plants to do something they would normally do, self-pollinate. This results in the F2 generation at the bottom of the picture. In the F2 generation, the white trait reappears. This was interesting because it meant that the white trait was still present in the F1 generation, but was just masked or hidden somehow. Mendel called these traits that were passed down over generations inheritance factors. We now call these alleles. It's important to remember that Mendel had a very strong mathematical background. He traced the inheritance of each of the individual traits in peas and kept very careful records of the numbers. He used his understanding of statistics and probability to make conclusions about how the traits in peas were inherited.
Okay, so alleles, that's another genetic term that we need to make sure that we talk about. We talked about a little bit with chromosomes, that we had an allele, one allele from the mom, one allele from the dad. So that's another word that we'll have to revisit as well.
Mendel published a paper in 1865 with these three conclusions to his research: the principle of dominance and recessiveness, the principle of segregation, and the principle of independent assortment. The principle of dominance and recessiveness states that one factor in any trait's pair may mask the effect of another. We examined this on the last diagram, when the traits from one generation reappear in later generations. The principle of segregation states that the two factors for a trait separate during the formation of eggs and sperm. Remember that this happens during meiosis, when the parents' homologs separate to make gametes. The principle of independent assortment stated that the factors for different traits are distributed to the reproductive cells independently of each other. This means that the offspring inherit a random mixture of the different traits that are present in their parents. At the time of Mendel's paper, chromosomes and the process of meiosis were unknown. Mendel based all of his conclusions on mathematics rather than an understanding of genes. Mendel's work was considered obscure and unimportant until the 1900s, when it was rediscovered and made important connections with the chromosome theory.
Okay, I'm going to stop here. If you're taking notes or if you have a place where you copy and paste and you need to know these three principles. So I'm going to give you a few minutes to write those down or place them in a folder where you can access them because some of the activities specifically ask you which principle is being expressed in this activity. So write those down so that you have something that you can refer back to. So I'll give you a few minutes to do that. And hopefully you have your screen up because I'm going to stop sharing for a minute so that I can make sure I get attendance done. And I'll come right back. But write those three principles down.
Mendel published a paper in 1865 with these three conclusions to his research. Modern Genetics Primer. Since the days of Mendel, a number of discoveries have been made that help us better explain.
Actually, I can't stop it.
Mendel published a paper in 1865 with these three conclusions to his research. The principle of dominance and recessiveness, the principle of segregation, and the principle of independent assortment. The principle of dominance and recessiveness states that one factor in any traits pair may mask the effect of another. We examined this on the last diagram. When the traits from one generation reappear in later generations.
Okay, mine is repeating because I switched
The principle of
screens.
segregation states that the two factors for a trait separate during the formation of eggs and sperm. Remember that this happens during meiosis when the parents' homologs separate to make gametes. The principle of independent assortment stated that the factors for different traits are distributed to the reproductive cells independently of each other. This means that the offspring inherit a random mixture of the different traits that are present in their parents. At the time of Mendel's paper, chromosomes and the process of meiosis were unknown. Mendel based all of his conclusions on mathematics rather than an understanding of genes. Mendel's work was considered obscure and unimportant until the 1900s, when it was rediscovered and made important connections with the chromosome theory.
All right, so we are talking about chromosome theory as well. There's a lot of things we have to
Modern Genetics Primer.
revisit.
Since the days of Mendel, a number of discoveries have been made that help us better explain what Mendel was witnessing in his experiments. Namely, we now know what DNA is, we know how meiosis works, and we can put these pieces together to come up with some vocabulary to describe the genetics behind Mendel's experiments. A phenotype is the external appearance of an organism that is inherited It refers to the physical appearance of the individual that is coded for in its genes. Phenotypes can be colors, as in Mendel's pea flowers, or a variety of possible patterns, as you can see in these bivalve shells, or even physiological effects, such as lactose intolerance. Lactose intolerance is caused by the lack of a functional enzyme that's supposed to be digesting the lactose sugar in milk. Remember that the outward appearance of traits, or the phenotype, is always coded for in the DNA.
Okay, so that's a vocabulary word that you have to be familiar with. There are two different types, genotype and phenotype. So you have to know the difference between the two, because you're going to, when we do the activities, you're always going to be asked two things. What is the phenotype and what is the genotype? So you need to know the difference between the two. Phenotype, think physical, and that is what we see. That is what is expressed. So when they say observable characteristic, what characteristic do we see? In your genes, we're going to see that we have one from mom and one from dad, but they both don't show up. So while you have one gene from mom and one from dad, you tend to look like one or the other, but the other one is still there. So what you look like or what is expressed is your phenotype. So make sure you write that vocabulary word down and have an understanding. I'll give yourself it is the inherited trait, but so is the genotype is also inherited. This one is the trait that you see. Okay, the visible expression of what is inherited. So she gave examples of the shell pattern. Are the swirls on the shell striped? Are they circular? Are they blended? What is the pattern on those seashells? That's a physical trait, the pattern on them. The color of the flowers, like we were looking at before, the color of the flower, the length of the stem, the wrinkle on the pea pod, all of those are what we will see. Okay. And then she talks about other characteristics like being lactose intolerance and other traits that we know are expressed that are not necessarily on the outside, but definitely have expression.
We now know that Mendel's factors are actually the genes that are inherited from mom and dad. We can also call these alleles. So for each trait, such as flower color or even tongue rolling, each offspring receives 2 copies, one from each parent. The combination of these alleles makes up the genotype of an individual, which we will talk about next.
Okay, so when we looked at chromosomes before, that whole structure is a chromosome. So we see we have one chromosome from mom and we have one chromosome from dad. So we have several names for the same thing. It's a chromosome, but it's also called an allele. Okay, and its specific alleles contains specific genes. So we also looked at the fact that humans have 23 pairs of chromosomes. Okay, and when we say pairs, there are 46 alleles. Okay, now think of an allele as a string of beads. Think of an allele as a string of colored beads, so that each bead on the allele codes for a different characteristic. So that whole allele from mom has several characteristics on it, and that whole allele from dad has several characteristics on it. But the ones that match up are the ones that match up for the same kind of trait. So like they have P identified on both of these. So say, for example, this particular allele has eyes and height and weight on it. Okay, so those alleles match up. And when they match up, as they go along, it'll decide which one is going to be expressed. So when we look at the gene map that we looked at before, and again, we'll look at it again, there were different size chromosomes and they all lined up to we call it genetic mapping when we take because they're not lined up like that in the cell, they're scattered all over the place, but we can line them up so that we match up the ones that are coding for the same characteristics. Okay, And so those alleles, this is what he studied, the ones that are the same allele for those particular traits. Okay. So again, allele is a term that you need to make sure that you understand. Allele is the chromosome that contains that trait from that parent. So you have a mom allele and a dad allele.
How can we represent alleles? Well, there are two types of alleles. The first is dominant, and those mask the expression of another trait. We will use capital letters in this course to denote a dominant allele. Recessive alleles are masked if a dominant trait is present. Remember that the white flower was masked by the purple trait. So the white trait is recessive, and we'll use small letters to denote recessive traits. If you have at least one copy of a gene that works, or a dominant trait, one that produces purple flowers or purple pigment, you can still produce the dominant trait. So you can see underneath the purple flower picture, we can make a combination of alleles that have at least one, but possibly two capital letters. These would both show the dominant purple trait. For a white flower, the only allele combination would be two small letters, or two recessive alleles to show the white phenotype, or outward appearance. The letter that we use to represent the gene doesn't have to be P. In fact, it's arbitrary. We could have used A if we wanted to. A couple other things that you need to know. Dominant genes don't necessarily suppress recessive genes. The recessive gene is usually an absence or a deficiency of some protein. Also, dominant genes aren't necessarily more common. Some of them actually cause disease, such as Huntington's disease.
Okay, so again, more vocabulary that you have to know. Dominant and recessive. All right, so dominant means that this is the characteristic that is expressed. In this particular example, flower color is what we're looking at, and the flower color that is dominant is purple. Now, she said it doesn't matter what letter you use, but they typically use the capital letter of the dominant trait. So this is why you have a capital P. Okay, and that's pretty much the rule. You can arbitrarily pick letters, but so that everybody stays on the same page, it is commonly expressed as whatever the dominant trait is, that's the capital letter you're going to use. Okay, so you need to know that dominant is expressed. Recessive is present but not expressed. It's like a tall person and a short person standing in front of the short person. The short person is there, but you can't see them because the tall person is blocking your view. So this is what we have with dominant and recessive. The gene is there, it's just not expressed because the dominant gene is more powerful. Okay, so again, the recessive gene takes on, in other words, white, we don't have a P and a W. Whatever the gene is for the dominant trait, that's the letter we're going to use, and we're going to capitalize the dominant trait. We're going to use the small letter for the recessive trait. So that's why you have a capital P and a lowercase p. Okay, and again, that represents one allele from mom, one allele from dad. So it could be with the two capital P's, both mom and dad. And right now we're talking about flowers, but both mom and dad flower had purple flowers, so they're capital P's. Second situation, one had a purple flower, the other had a white flower, and the gene doesn't disappear, but it's not, the lowercase is not expressed. So you have capital P and a small p, but when we look at that, and we talked about phenotypes, when we look at that flower, it is purple. Okay, the only way we get a white flower, only way that a recessive characteristic is expressed if both genes are recessive. So that means mom gave a recessive gene and dad gave a recessive gene. And if they're both lowercase, then we get a white flower. Okay, so whatever characteristic is recessive only shows up if both parents give that gene. All right, so again, make sure that you, so this one little box is giving you a wealth of vocabulary, and we're going to take each of these separately, but just know that in this one little presentation alone, it's the basics of a lot of what we're about to do.
The genotype is the genetic makeup of an organism. It refers to the letters or the alleles that an individual received at fertilization, one letter or one allele from each of its parents. The purple flower has two possible genotypes. It could have two capital letters to make up one genotype, or a capital and a small letter. The white flower has only one possible genotype. It would be made up of two small letters.
Okay, so the first word I told you was phenotype, and that's what you see. All right, the second word now is genotype. So what genes are present? Okay, so we're going to, when we do the activity, we always map out what genes are present. And after we map out what genes are present, then we have to decide what are we seeing based on the genes that are there. So again, when we say the genetic makeup, we're saying capital P, capital P. So that is, both of these are dominant for the purple flowers, or it's capital P, small p, which means that we have one parent that gave a purple gene and the other parent gave a white gene. Or we have small p, small p. So when we express genotype, we're only talking about the letters. Okay, so we would say, if somebody asks you, what is the genotype here? You'd say capital P, capital P. Or you would write capital P, or capital P, small p, or small p, small p. That's how you write genotype. If they ask you what is the phenotype, then you now have to express what do those letters mean? Okay, so phenotype tells us what is an interpretation of what these letters are. All right, so now we have two words, genotype and phenotype.
There are three kinds of genotypes. The first is homozygous dominant. In this genotype, there are two dominant alleles for a trait, such as capital P to represent purple flowers. The homozygous recessive genotype would be a genotype that's made up of two small letters, or two recessive alleles for a trait, like you would see in white flowers. Finally, there's a heterozygous genotype. In this genotype, you would see one of each type of allele for a particular trait. So you would see a capital and a small letter in combination.
Okay, so here's some more vocabulary. I tell you, there's a lot of vocabulary with this that you have to distinguish the difference between. So the term homozygous has two ways to be expressed. Homozygous means same. So just know that what that root word means. Homo means same. So homozygous, you can be dominant. That means both alleles are the same. And homozygous recessive, that means one allele is different to the other allele. Okay. Actually, sorry. Homozygous recessive is they're the same, but they're recessive. They're the lowercase letter. Okay. Heterozygous is they're different. So you have one dominant, one recessive. Okay. So hetero means different. Homozygous means the same. So homozygous can be dominant, your two capital letters, or it can be homozygous recessive, your two lowercase letters. So again, a lot of vocabulary.
Finally, let's apply the terminology we now understand to Mendel's work with pea plants. You can see in the purple box what we do know. White flowers are recessive to purple. Therefore, their genotype is small p, small p. Purple flowers are dominant and must contain at least one dominant or capital letter. Their genotype would be big P, or big P, little p.
Okay, so again, that's a rule. That's a rule that you have to understand. So what we call this is cross-pollination. We are taking one type of plant and combining it with another type of plant. So they're taking the plant that has white flowers, which is recessive. So it has two small P's. And we are crossing it. So you have that little X in the middle. That means we are crossing it or fertilizing it with a plant that has purple flowers, and that's dominant. So they have two capital P's. Now notice in this generation, because they only have those things to give. So the white flower is going to give a small p and the purple flower is going to give a capital P. So look at our F1 generation. Our F1 generation can only be heterozygous because it has to get one from mom, it has to get one from dad. And so the only possible way the first generation can be, the only possible outcome for the first generation is to be 100% heterozygous. Okay? All right. Now, when you do the second generation, if you take those two, now we look at the possibility that one parent can give a capital P or it can give a recessive P. And the same thing with the other parent. So when you look at the second generation, the rule for that generation is always going to be, and then we're going to look at, we call it possibilities, because when we don't know which egg is going to meet which sperm, so the possibilities, and when we look at 100%, we say 25% there, 25% this. So like notice I said 100% in the F1 generation. So we're going to talk about this in terms of percentage. When we look at the second generation, we see there are three different plants that we're going to turn up purple, and one plant that's going to turn up white. So we're again looking at this a couple of ways. We can say that it's a 75% chance that the plants will be purple and 25% chance that the plant will be white flowered. Also, with our 75%, they don't have to be homozygous. They can be heterozygous because they can be a capital P, small p. And we're going to look at the possibilities of that. So we have a lot to unpack with just doing this activity. So we're going to spend quite a bit of time doing that, but it is important that you spend a little bit of time understanding the vocabulary so that while we're pulling these maps apart, that you understand, what am I looking for? What do I mean by homozygous? What do I mean by heterozygous? What I mean by F1 generation or the parental generation? So you need to go back and look at your vocabulary in order to do these activities.
Recall from earlier that I told you in the P generation that Mendel used purebred plants. This means that the white flowers must have been homozygous recessive, or small p, small p. The purple flowers then must have been homozygous dominant, or big P, big P.
All right. And that was an important observation because again, like I showed you in the F2 generation, it's a possibility that it could have been a capital P, lowercase p, in which case that gives you a whole different outcome. And we will look at that because we're going to look at different generations and what the outcomes will be.
Each of the parents in the P generation donated one of their letters to the F1 generation. This resulted in a heterozygous dominant genotype, or a genotype in which you had a big and a small letter.
All right, we got 1 minute left. So I'm going to stop it here. But again, for tomorrow, we're going to just spend time going back over this particular section because again, a lot of this information is necessary to understand before we do any activity. Okay. All right, any questions? Any questions? So again, you see with this one, we hadn't even gotten past the anything yet. We hadn't even finished this yet. So this is this one little box right here. Make sure you go back and look at all of this again, because there's a lot of vocabulary just in this box right here.
How can we represent alleles?
Oops. Okay, so I'll see you all tomorrow.
Gregor Mendel is known as the father of modern
Hopefully it'll stay with me.
genetics primer, Mendel and modern genetics terms.
Stop sharing.